molecules that are dissolved in arterial blood, not atmospheric PO2 or alveolar PO2, but arterial PO2. So the x-axis is a horizontal axis. Okay? Remember that was our arterial PO2, and then this is the y-axis, and those values are determined by measuring
Hemoglobin saturation.
okay, percent hemoglobin saturation. All right, and I said there were two significant areas of that oxygen dissociation curve, and what was their significance? Remember, if we diagram this, it's a curve that's in the name. So the two areas are this region, which is known as the plateau, and this region, which is the steep portion of the curve, or just the steep gradient. All right, so what is the significance of those two components? So the plateau shows that for a wide range of PO2, we can get 90 or over percent saturation of our hemoglobin. We don't have to have a PO2 of 100 or higher to get 100% saturation, okay? We can get, in fact, our chemoreceptors in the aorta are not sensitive to trigger a deeper breath with low oxygen until the PO2 drops below 60. So 80 or higher, we can get 90% or more percent saturation. So this provides a wide range of oxygenation. That's why we can have individuals living in Mexico City or Denver or Lake Tahoe and still be able to function normally. Okay? The significance of the steep gradient.
Detachment of oxygen.
That's right. It's the oxygen now will dissociate from the iron atom of the hemoglobin and will move to the tissues. And so again, we see that happening. The most important region is between 40 and 60 or 30 and 60 millimeters of mercury. And so this is lung involvement and tissue involvement. All right? So what did we mean when we said, or I said, what did I mean? When we talked about the curve shifting to the right.
Oxygen comes off the hemoglobin easier.
Okay, so we have decreased the attraction. Anything that decreases the attraction between oxygen and iron, so the oxygen in the heme portion, will cause that curve to shift to the right. So there's a decrease in the affinity. So anytime we have a shift to the right, there is, and that's not showing up very well, there is a decrease in the affinity of between iron and oxygen. So what types of changes?
Increase in temperature, increase of CO2,
Okay, so increase in temperature,
increase the hydrogen ions.
increase in CO2, and an increase in CO2 can do that through two mechanisms, one direct and one indirect. So what is the direct way, or the more hand-to-hand combat way? What's the direct way in which carbon dioxide decreases the affinity between iron and oxygen?
The hemoglobin.
Binding to it
hemoglobin.
directly, all right? So as carbaminohemoglobin is going to have a direct effect binding to the end of one of the amino acid residues, and again changing the structure of the hemoglobin molecule, so it is more difficult for oxygen to get into or to remain associated with the iron. And the indirect effect?
Increase the flow acidity.
Increasing acidity. So increasing the hydrogen ion concentration via, remember we ended up in the last 15 or so minutes of class on Friday by talking about the formation of carbonic acid.
Is there an extra A in carbamino?
No,
Okay.
carob, I'm thinking of chocolate. And so these hydrogen ions can affect the pH. And again, extra hydrogen ions bind to the globin and change its structure. Is that our only source of extra hydrogen ions?
Decreases pH.
Anything, that's the respiratory cause. So when we do our urinary system, the last lecture is on acid-base balance. And we'll talk about metabolic causes. So someone who has diabetes, it is breaking down lipids and increases the number of fatty acids, is going to have an increase in hydrogen ions. Someone who, I mentioned this to the class this morning, someone who has severe diarrhea, the pancreas, as we'll see when we get to the digestive system, releases bicarbonate ions to buffer the hydrochloric acid that is released by the stomach. With diarrhea, those bicarbonate ions are not absorbed into the body, and we lose that buffering capacity. So someone who has been really sick with diarrhea, coming back from Mexico, the spring break or whatever, may have a high amount of hydrogen ions even though they have a normal CO2. Okay? And so it doesn't have to be the hydrogen ions from CO2, but CO2 can affect hemoglobin binding through those two effects, either directly in the formation of carbaminohemoglobin or the production of hydrogen ions from carbonic acid. All right, and we also have the 2,3 BPG or DPG. All right, so then if that change of those changes made a decrease start in the affinity, then a shift to the left is going to mean an increase. In affinity between iron and oxygen. And with a couple of exceptions, all we need to do is just do the reverse of this. So a decrease in temperature, a decrease in CO2, a decrease in hydrogen ion concentration, a decrease in 2,3 DPG, all right? So reverse of all of those will cause an increase in the affinity. But we added two additional factors, carbon monoxide
And fetal hemoglobin.
and fetal hemoglobin. Okay, so you remember the picture of the chair I drew? So, if you're playing musical chairs, we would have a chair with a steep angle, very hard to sit on, so the oxygen has a difficult time binding, okay? And if we were to draw the chair over here, you'd have your bottom stuck in that area there, it'd be hard to climb out of it. You're going to be in that chair longer. Okay. Any questions about carbon dioxide transport? Yes.
I have a question about the fetal
Sure. Go
hemoglobin.
ahead.
I don't understand how it works.
Okay.
What's it actually do?
Fetal hemoglobin has the alpha chain, but instead of the beta chain, there's a couple of changes in amino acid, histidine, serine instead of histidine. It's called a gamma chain. Okay. Because of that, its interaction with oxygen is different, and also it doesn't interact with the 2,3 DPG, so it holds on to the oxygen more tightly. It's more that crack form in the sense that it's more difficult for the oxygen to be released. It's not released as often, so at any given time, you're more likely to have oxygen bound to the hemoglobin. And so it gives it a higher affinity for oxygen than the adult hemoglobin in the maternal blood. So when it diffuses into even at a lower PO2, when it diffuses into fetal blood, and binds to the hemoglobin, it's not as likely to come off and go back to maternal blood. So it allows, it shifts the transfer of oxygen to the fetus.
And that will affect both mom and the child?
Yeah, so as the oxygen shifts from the mom to the fetus, then obviously the tissue PO2 in the fetus is going to be lower too, to bring that oxygen off because of that higher PO2, the higher affinity. So the fetal tissue will have a lower PO2 before the oxygen actually comes off. Does that help?
Yeah.
Okay. All right, so anything else? Any questions on the carbon dioxide transport and the carbonic acid? This morning's class, a student asked about the relationship, the term shift, from our oxygen dissociation curve, the right and the left shift. She had confused with the chloride shift and wondered how the chloride affected the oxygen dissociation. Was that you? Or Shamika? Okay. So I don't want you confusing the term shift. It's talking about two different components. So when we were talking about the red blood cell, and remember we formed our carbonic acid, and then some of that would dissociate to form a hydrogen ion and a bicarbonate ion. As we'll see with the urinate system, bicarbonate ions are a very good chemical buffer. With high hydrogen levels, hydrogen will go back to and bind to the bicarbonate ion. All right, because remember this is concentration driven. So that becomes helpful in buffering the pH of the blood, so the bicarbonate ion moves into the plasma from the red blood cell, but to balance positive and negative charges, chloride ions from the plasma move into the red blood cell. And now we have the same number of negative charges as before, but the bicarbonate ion is a better buffer. And so this is known as the chloride shift. So it's an entirely different concept than the right or left-sided shift that we talk about with the oxygen dissociation curve. All right, so then, in talking about this, what does the initial CA stand for? What do the initial CA stand for?
Is that the enzyme?
It's an enzyme.
Carbonic and
Carboxylates. Okay, so that's an enzyme that's present within the red blood cells, and it increases the rate of carbonic acid formation. So that's going to occur. Carbonic acid is going to form in the plasma. We obviously have a lot of water in the plasma. So CO2 in the plasma of the blood will form carbonic acid, but more of it will form in the red blood cell because of the presence of carbonic anhydrase. And as we'll see today in the regulation of breathing, we have carbonic anhydrase present in cerebrospinal fluid as well. And so we have a higher rate of carbonic acid formation there in cerebrospinal fluid. All right, so moving over, I was going to say the word shift, but that might be an overuse of the word. Moving to the control of respiration on page 70-72. So here's a question to kind of prime your thinking process. Which of the following chemicals is the primary central nervous system factor in stimulating a change in respiratory rate or depth? Okay, A, arterial PCO2. Yes, to a point. Now what if I change the question to, which of the following chemicals binds to CNS chemoreceptors to cause a change in respiratory rate or depth? It starts with A, but carbon dioxide does not bind to the chemoreceptors, the central nervous system chemoreceptors. Hydrogen ions do. C or D? D.
Because the O2 doesn't allow for O2 to constitute.
Arterial pH hydrogen ions are significant and certainly change your respiratory rate, but their receptors, as we'll see a little bit later, are in the aortic arch or in the carotid sinuses. The hydrogen ions present in the arterial blood as it passes through the brainstem, remember the blood-brain barrier? So because of the blood-brain barrier, those hydrogen ions, unless they're in very, very high concentration, do not leave the blood. It is the CO2 that leaves the blood. And then, as we see in that diagram there with the red blood cell, in the cerebrospinal fluid, forms carbonic acid and dissociates to form hydrogen ions. So in the end result, it is arterial PCO2. As it rises or falls, it changes our respiratory rate, but what actually binds to the central chemoreceptors is the CSF hydrogen ions formed from that CO2. Okay? Okay. So,
That's 70/30.
oh, is that what it is? Okay. So when we look at the control of respiration, we have within the brain stem, we have the central controller. So we can actually take an individual or an animal and remove the brain at the level of the midbrain, and they would still breathe. Okay? Those of you that had me last semester, and I talked about one of the last weekends, I worked as an RN in labor and delivery. We had a woman come in through the emergency room who was pregnant without prenatal care, and she was in labor. And the OB that was down, the ER was called in down to the ER, sent her up to the labor and delivery floor. And another nurse and I checked her. The baby's heart rate was 90 beats per minute, which is kind of on the low side. Range is 90 to 160. And then it started dropping. So we called an emergency cesarean section. They don't worry about a bikini kind of a cut. The same OB that was in the OR came up along with pediatricians. I worked with the pediatrician at the little bassinet, and they put the mother immediately into unconsciousness without the epidural or anything. They just gave her general anesthesia and brought the baby out. And the baby had little flippers for upper extremities and little flippers for lower extremities. And when they brought him over to the warmer, the back of his head was kind of slanted like this. But he was still breathing, and he had a heartbeat. It lasted for about 5 minutes before he stopped breathing. Earlier in that summer, we had a similar baby that lived for about 2 hours. Okay? So they're called mare anencephalic. You don't have to remember that term, but the babies are born without much of a cerebral cortex. Instead of the full gyri and sulci, it's basically just a flat membrane. I did read of one case where the child was not expected to live more than a few days, and at the age of 3, she was walking and talking and running around, and she developed that cerebral cortex after birth.
Wow.
Which doesn't happen. I mean, it was an unexplained event. And so those babies, though, were still breathing. And in Canada, which has, well, I don't know what's going to happen to our health insurance, but in Canada, the government pays for fetal heart transplants. And so when my sister worked at Loma Linda at the pediatric heart transplant, a lot of their patients came from Canada. And the baby that needed the heart transplant would come, and the mother carrying the mare anencephalic child would come, and they had already decided to be organ donors. And so when their babies were born, they were still breathing, and the heart was still beating, because those centers for cardiovascular and respiratory control are in the brainstem. And I don't know if they waited for the baby to stop breathing, or if they had the parents' permission to, then you would have compromised heart tissue, but they waited until the heart rate dropped to a certain level. I'm not sure how they decided, but those were the organ donors for the hearts for the babies that needed them. So that central controller that is in the medulla, and if you still have the medulla, at least at a minimum, the baby is still going to breathe. All right. It might be a kind of a very irregular gasping type of breath if they don't have the pons, but they will still have the breathing. All right, then we have the sensors. So in lab today, you're going to be seeing how long you can hold your breath, what the effect is if you hyperventilate, or if you take in deeper breaths, and of course then the motor responses are the effectors. So this is a reflex. You do not have to have the cortex to breathe, because the source of activation of the lower motor neurons is in the brainstem, okay? So here I'm going to kind of diagram a similar part of this over on this side of the board, so let me do that here. Note the relationship between the fourth ventricle and the medulla, because the fourth ventricle, this fluid-filled space here, is where the CSF is, that we're talking about changing with the amount of CO2 that's available. The fourth ventricle and CSF. Medulla oblongata. And the pons. So the medulla oblongata, the pons, even cerebral cortical areas are going to be where the central components are. And then our respiratory muscles are skeletal muscles, but they are activated via reflexes. So I'm going to talk about the three components of neural control of respiration. So the first one is, it used to be thought that that generator, that separate generator, didn't occur. Remember how in the heart we had a pacemaker? And we had modified muscle cells that depolarized on their own and sent the current to our cardiac muscle. All right, so the equivalent of this would be that the skeletal muscle, our diaphragm, would just depolarize on its own, and every 5 seconds would reach a voltage-gated threshold and open the channels, and our diaphragm and our external intercostal muscles would contract. Well, we don't have that, okay? Then we wouldn't even need the brainstem to breathe. But then the thought was, well, the collection of neurons, the nuclei of neurons that send fibers, nerve fibers down to those respiratory muscles, the dorsal respiratory group, maybe that's where the cycling occurs. And now the thought is, well, no, there is some evidence, mostly seen in mice or rats, that there is a structure in the brainstem that is, and it's controversial whether it's a cluster or a network, and that's neither here nor there for us. But there is a group of neurons shown in red here. This would be the fourth ventricle, looking at the dorsal view of the brainstem. And these cells depolarize and kind of set up a pattern generator. So if we go back, so the generation of the respiration rhythm now is thought by most individuals to be due to this structure called the pre-Botzinger complex. All right, so I'm going to diagram this in here, and we have, I'm going to go ahead and put the multiple groups of cell bodies. What I'm ignoring that you might read about if you were to read college level text on this is the nuclei for the trigeminal nerve and the hypoglossal nerve and the vagus nerve and glossopharyngeal nerve and all of those components that are also considered to be important. But back here, we have a group of nuclei known as, nuclei being cell bodies, known as the dorsal respiratory group, or DRG, and I'm going to come back and talk about that in more detail. It's dorsal because it's on the dorsal surface of the brainstem. And we have another group known as the ventral respiratory group, or VRG. And part of that VRG is this pre-Botzinger complex. It's just a fun phrase to say. So that is thought to be a group of neurons that depolarize on their own, similar to the way in which the SA node or the atrioventricular node had leaky channels and would just depolarize on its own. Okay? And that seems to be the region that sets up the neurons that fire and send excitation down to our diaphragm. All right? Kind of the pattern generator. But it itself doesn't directly send neurons to the muscles of inspiration. So what I've shown here in red would be the pre-Botzinger complex. The yellow would be the rest of the ventral respiratory group. And then this would be the dorsal respiratory group. So I copied this, I drew this from another diagram that had about 10 other nuclei on there, which was way too distracting for you guys. And then that's a sagittal view. So then we have the regulation of rate and depth of breathing, or the determination of rate and depth of breathing. And this is what you're going to experiment during lab today with the breath holding and hyperventilation, right? So here we have a similar sagittal view. This would be the fourth ventricle. Here's the dorsal respiratory group, and here's the ventral respiratory group, all right? And we'll see how that's going to determine inspiration, or if we change the amount of CO2 in the blood. So our normal inspiration is 2 seconds in, 3 seconds out, 2 seconds in, 3 seconds out, about 12 times a minute. Every 5 seconds we take a breath, all right? If we, one of the things we're going to do today is to breathe, rebreathe your CO2. Got to give it to your child. So I'm rebreathing CO2. So as CO2 is elevated in my blood, would I be able to hold my breath for the same amount of time, longer or shorter? Shorter. It should be shorter, okay? And that's the effect that we'll see in a little bit of elevated CO2 on the dorsal respiratory group. The ventral respiratory group is going to modify changes to that. So can I breathe harder? Can I put a force air out? Could I suck air in? So that's the changes that so the ventral respiratory group, is affecting those changes. And then the third thing is the modification of respiratory movements. So I have an irritant and I cough, or I laugh, or I sneeze, or I talk. And those things are going to modify further other than just chemicals. All right, so when I was reviewing and preparing for this lecture over spring break, hypoglossal nerve kept popping up as being affected. Like, hypoglossal nerve doesn't go to the lungs, hypoglossal nerve doesn't go into any viscera, but speech is going to modify your respiratory patterns. Okay, so movement of the tongue, swallowing. And we move the tongue up and towards the back of the pharynx when we swallow. So those movements of the tongue affect changes in respiratory movements. So these are the three things that are involved in the control of respiration: the generation of our pattern, or just a general pattern, the regulation of the rate and depth, and then modifying that because of voluntary or emotional changes in the body. Okay, so now let's come to this. I'm not going to go through, it's kind of a summary of what I'm going to say. I just put it on one slide, and I'll upload this. I made some changes to what's already on D2L. So we start breathing. Okay, I'm just going to go through the pattern, and we'll come back and look at the specific nuclei. So we start inspiration, and as we'll see in a few minutes, this is the dorsal respiratory group involved in that. The dorsal respiratory group fires, and that will activate the phrenic nerve and the intercostal nerves. And as we'll see also, there's a ramp effect in that, increasing firing, more and more muscles and fibers involved as the diaphragm lowers even more and the ribs lift up even more. And that lasts for about 2 seconds. and then all that stops. So the inspiratory neurons in the dorsal respiratory group stop firing, and without stimulation, the diaphragm gradually relaxes and moves back up. The intercostal muscles relax and move down, and expiration lasts for about 3 seconds. Remember, we said that was a passive process. So what we have involved here, other than the pattern generator, is the dorsal respiratory group. So it is dorsal respiratory group, which has only inspiratory neurons, that's involved with quiet breathing. Okay, so dorsal respiratory group is shown by the second cluster right here, and shown right here.
Can you do that again?
Yeah. So dorsal respiratory group is shown right here. Okay, and I'm going to do a bunch of writing that's on the lower part. I should have moved all this up, but anyway, didn't learn my lesson this morning. So when those dorsal respiratory neurons fire, and we have that ramp effect, they send, this is kind of the equivalent of our upper motor neuron. So if I want to move my finger, if you guys remember from last semester, it's a 2-neuron pathway. Cell body and the precentral gyrus fires. That neuron passes down through my pons, through the cerebral peduncles, the pons, the medullary pyramids, and goes down to the cervical portion of my spinal cord and interacts with the anterior horn cell bodies of my lower motor neuron that then goes out to my extensor muscles, and I extend my finger. Okay? So it's a 2-neuron pathway. Breathing is a 2-neuron pathway as well. And if we're not involving the cortex, then the equivalent of the upper motor neuron is the inspiratory neuron cell bodies in the dorsal root ganglia. So dorsal root group. So let me draw those in. So that is going to then go down the spinal cord to the anterior gray horn, and the cell body will then go out to the phrenic nerve. Go out to the diaphragm. So this would be the phrenic nerve.
I got that right in my class.
And as well, we would have neurons of the dorsal respiratory group that go farther down the spinal cord, and their lower motor neurons would go to the intercostal muscles. And which intercostal muscles would these be for inspiration?
Internal.
The external or the internal?
Internal.
Inspiration. External. The fibers that form the V. So these are going to go to external intercostal muscles. And the nerves would be the intercostal nerves. And I'll let you guys come up here and take pictures if you want in just a bit. So when those fire, they fire for 2 seconds with an increasing number of nerve fibers, innervating an increased number of muscle cells, and I breathe in. All right? So the dorsal respiratory group is formed of inspiratory neurons. I stands for inspiratory, not for inhibitory. Okay? So inspiratory neurons, they fire in a ramp effect, and then you breathe out as soon as they stop firing. So we're not activating expiration muscles here. Expiration occurs because these fibers no longer fire. The ramp effect is referring to an increasing number of nerve fibers firing, and so we see this spiking that occurs, shown here. So, the ramp effect, notice what happens to tidal volume. It goes up, we reach the peak, the neurons stop firing, and expiration occurs. Okay? So we see this in our 2-second inspiration and our 3-second expiration. 2-second inspiration and 3-second expiration. If we have the pons information smoothing this out. If all we have is the dorsal respiratory group, then it doesn't look like this, okay? But with normal passive breathing, with both the pons and the medulla working, that's the pattern that we see. Okay, but it takes the pons to smooth that out. Now, let's say